as usual, a couple of quick announcements. The first one is here in the middle, Portland Festival, Winter Festival of Lights. I don't know how many of you know about this, but it's really cool. I'm a big fan of STEAM, of science, technology, education, arts, and math. Over at OMSI, they have this amazing fire-breathing dragon. So I highly recommend going over there and checking it out. Also over in the Shattuck Annex, some of the art students, I believe, have some projects they'll be presenting over there starting, I think, this afternoon. So highly recommend checking that out. We will be talking about fun supercoiling things later, so we will get to that. But probably most of you are interested about what happened on Wednesday. I did post all of the scores, and my exams are getting better, whether you think so or not, because I'm actually getting a distribution that's getting close to 50. In my perfect world, the high score would be a 50 out of 50. Somebody got a 49, you know who you are, and I know who you are too, but don't necessarily show that to everyone. But one thing that I have asked the people who've gotten high scores to do is to please let me know how they did it, and I will share that. So the basic idea, since I'm doing a straight scale, if everybody gets 100, everybody gets 100, everybody gets an A. Yeah?
How, in what mathematical world is 30 to be?
But in what mathematical world? That's the number of points. So basically, as I listed in the syllabus and also in lecture 1, I set the high score to 100%, but if I'd done that with 49, then lots of people would be getting lower scores. The 49 was an outlier, but there were still 7 people who ended up with more than 44 points out of 50. So, in theory, I could have just scaled up to there. But again, these are outliers. So I'm scaling to 44, and you can do 90% of that's an A, and so on and so forth. I only assign letter grades at the very, very end of the course. So I'm going to add all the numbers together, your clicker scores, all your exam scores, etc., normalize at that point, and I will tell you, I'll post it on D2L exactly what that is. So if you are somewhere below this 19 number, pardon me, then we should probably be talking, or at the very least emailing, about how to do better as far as is concerned. Because I kind of feel that if people are getting scores lower than this, I'm somehow failing. So that's how I feel about it. There are a couple of fail questions, I guess. What I always do after exams is I go through and look at the questions that the most people missed. So if you got these right, think yourself very in tune or lucky, as the case may be. A couple of different things here. So by definition, a multimeric protein has multiple mains, more than one N-terminal amino acid, multiple motifs, beta sheets, alpha helices. Most people selected multiple domains. Domain is by definition a folded structure. You don't necessarily have to have a folded structure. This is one of the examples I pulled out from the textbook. This does not have a folded structure. It only has a folded structure when it's together with this one. And so here, you don't necessarily have multiple domains. You do necessarily have more than one N-terminal amino acid because you have more than one protein. Anytime you have a multimer, it's multiple polypeptides. And I guess I didn't do a good enough job of explaining that. I'll talk about multimers and subunits quite a bit as we move through the rest of the class. So wanted to make sure that was clear. The second one has to do with 5' and 3', and DNA polymerases and templates and so on and so forth. So if you look here, here's your polymerization reaction without the DNA polymerase. Here's your template strand. Here's the primer strand that is getting extended. The polymerization is always from 5' to 3'. You keep adding on here. But that means the polymerase is moving in this direction. What direction is that relative to the template strand? It's 3' to 5'. Yes, no, I see some nods, I see some shakes. So, and in fact, believe it or not, I thought that most people were going to give me a hard time about this because of E, which I think absolutely nobody picked. Because if you're talking about proofreading, you could maybe think about it going backwards. So, if you actually look at the polymerase, we talked about this really, really briefly. It's just the template that's moving, and actually not the template, it's the new strand which is moving. The polymerase actually doesn't move at all during the proofreading reaction.
So if you ask which way it's synthesizing, then it would have been the 5' and 3'. But because it's which way it's moving, not what way it's. Okay.
Yeah, so I'll repeat your question here. If I had asked, which I didn't, how it was polymerizing, that definitely would have been from 5 to 3. Or if I'd asked how it was moving relative to the non-template strand, that would be 5 to 3. And now we're going to spend a lot of time talking about 5s and 3s and polarities. And as if we haven't already, there'll be more of this. And this is kind of why I'm beating on this particular theme here. Yeah? Why? Why did I ask the question that way? Because I don't want people to memorize facts. I want you to think. No, I want you to think. I'm sorry if I'm asking you to think. But sorry, I'm not trying to pick on you personally. No, my reasoning is I want you to understand it more than a fact memorization. That's my reasoning for that. Yeah, one thing that, okay, I think I've mentioned this to a few people in my office hours already. Be very careful about reading questions. And I had a bunch of people asking me about, isn't this the same answer from the nucleotide language to amino acid language, amino acid to nucleotide language? They were different. They were opposites. And just be very careful. You've got plenty of time. I don't give you a time limit for this. So please take your time. You've noticed a lot of people did ask me questions. And I tried to answer them. So next time I expect I'll be running around the whole time during the midterm. Okay, I will bring back your scantrons. I haven't alphabetized them yet. If there is a question, like maybe you think the scantron was misscanned, let me know. I've got them all literally scanned into PDF, so I can look at those. So if you think there was an issue there, please let me know. Okay, more midterm questions. Yeah?
So I know you said you're going to compile all those scores at the end to give us our final grade.
Mm-hmm.
But for this midterm, are you going to update them so they're not out of 49 anymore, they're out of 44? Okay, so the percentages that D2L gives you, I have lots of issues with D2L. So I'm trying to answer your question here. The way that it gets uploaded, if you upload the scores and had something higher than their score that they have, then it actually will cut off any score that's higher than that to that particular score. And so I just take the total scores across the board. And so that, those numbers don't really make sense. So don't look at the percentages in D2L. It does that automatically. And there may be a way to switch it. I'm just not quite sure what it is. If somebody knows, please let me know. And then also, just really quick, because you said if you got 39 or above, that's an A. But 39 out of 44, isn't that A-?
Well, you could, again, the different calculations, et cetera. Yeah, the math is such, and I could put out the X number of decimal points here. I will use those calculations at the end. This just gives you a bit of an idea where you stand.
Yeah, like 30 divided by 44 is like 68 point something.
Mm-hmm.
Definitely not a B, which is why I was confused.
No, above 60 would be a B. I think 60 to 70, I forget what it is. But again, if people want to talk about that, I'm happy to show you these numbers. And I may have miscopied them when I looked at the normalizations. But if you take 44, back up here. This thing is still not working. My mic is running away on me here. Take 44, multiply by 0.9. If you have a higher score than that, you're looking at an A. Again, I don't assign letter grades until the very end of the class. But that's a start, and then I forget what all those other numbers are. You can see them on my PowerPoint and on the syllabus. You can do those calculations. And if you're off by like a 0.2% or 0.2 in terms of a number, or 0.2%, at the very end, I will definitely look at those. These are your minimums. So if you've got this and above, I'd say you should be pretty happy. If you're really borderline, then do better on the next couple of exams. And if you're still borderline at that end, we'll see what happens at the very end. If anything, I usually end up going lower than these scores in terms of final letter grades. OK, now let's talk about interesting stuff, like replication and DNA repair, et cetera. So we got through, and someone asked me right before class, how much are we going to cover, skip, et cetera. We will finish up with DNA replication. There are a couple of important things that I wanted to talk about, and then move along to DNA repair. So covered everything up through the replosome. We haven't really talked about eukaryotes other than the nomenclature. The eukaryotic replication fork is a little different than the bacterial replication fork. I wanted to point out some of those differences. Then supercoiling, which, again, is a subject we could spend all term talking about, but we'll go through this really briefly, and that's why I've got my phone cord here. Yes, phones used to have cords. And then a little bit about how replication starts, because so far we've been talking about as replication is continuing. And the start of replication is an interesting issue. And then finally, what happens at the very ends of chromosomes. Linear chromosomes have a really serious problem due to the fact that you're starting with RNA, and that RNA needs to be removed a little bit later on. So let's look at the eukaryotic replication fork. A couple of important things here. The leading strand is basically identical to the leading strand that you see in bacteria. You've got a replicative RNA polymerase. You've got a sliding clamp. You've got single-stranded binding proteins and a helicase. The helicase here in this particular image is listed as T antigen. That's not the case that was in a virus-infected cell, how it was originally found out about. This is really MCM and the GINS complex. Exactly what those are for is not particularly critical. What's much different as far as bacteria and eukarya are concerned is what's going on in the lagging strand. So the lagging strand also has a DNA primase, which is an RNA polymerase, which makes RNA. But that's always in complex with a DNA polymerase, so it makes DNA, called Pol alpha. So people always talk about the primase polymerase complex on the lagging strand, and it starts also on the leading strand when you do initiation. We'll talk about that in just a second. So it's this primase polymerase together that do this. So the primase puts on a little bit of RNA, DNA Pol alpha extends that a little bit, and then that whole complex comes off, and then you have the replicative polymerase, which in eukaryotes is Pol delta, Pol epsilon. So that's one difference. We've got this Pol alpha here. The other difference is that, this is Pol alpha. It's not DNA polymerase 1. DNA polymerase 1, E. coli, the best organism ever, depending on who you talk to, that has, as well as its 3 to 5 perforating activity, it also has this 5 to 3 primer, degrading activity. Eukaryotes don't have that. So eukaryotes have to have another way of dealing with primers, getting rid of primers, and getting rid of the RNA. One of those is RNase H, which is just the same, well, activity-wise, just the same as you have in bacteria. But then there's this thing called Fen1. What's Fen1? Fen1 is the flap endonuclease. So what's a flap? A flap is that RNA which has gotten peeled off by the replicative polymerase, which is coming along and making DNA. So you have a flap of RNA. This flap of RNA gets cut off by an endonuclease. So exonucleases come in at ends, endonucleases come in the middle. And so the flap endonuclease will cut off that primer. So in terms of function, DNA polymerase 1 in bacteria can chew up through an exonuclease activity the primers. In eukaryotes, the flap endonuclease cuts in the middle, and that piece is removed. So another thing that you may have noticed on here that we haven't talked about yet is this topoisomerase. What the heck is a topoisomerase? Changes the topological form of DNA, of course. Well, why do you need a topoisomerase? Basically, what topoisomerases are involved in is changing the coiling of DNA. Why do you need to do that? Well, that's because, ooh, interesting stripes on here. Is that my computer or is it this computer? It's this computer. OK. Shouldn't be important. So what happens if you pull the two strands of DNA apart? What's doing that? The helicase. So the helicase is pulling these two strands apart. But these two strands are normally wrapped around each other. So if you pull strands of DNA apart, what happens is the rest of the DNA starts to coil on itself. And this is just the fact that you're wrapping the DNA around. Each time you wrap, you're unwrapping, and that wrap is always going to be conserved. And so this is just a real quick example over here. Here I have my phone cord. So it's wrapped in one particular way. If you unwrap it, so I'm just taking this and twisting it in the opposite direction, what happens? It starts to wrap around itself. And that's one of the reasons we don't have phone cords. But also, that's exactly what's happening with DNA polymerization, is you're unwrapping it. And the other thing that you can't tell, but the more you twist this, the harder and harder it gets to twist. Again, not surprisingly. And so what's going to happen eventually is the polymerase is going to get stuck. It can't move through here. Actually, more importantly, the helicase, it can't unwrap those strands anymore. You can't keep going. It just gets to this point that everything is stuck. So how do you deal with that? Topoisomerases. So the topoisomerase basically gets rid of these extra loops. So this is just an example up here. Here's our replication machinery. Basically, again, this is the helicase generating what people call supercoils. So the coil, the normal coil, is just the two strands that are wrapped around each other. Supercoiling is when you have these strands wrapped around each other. So double strands wrapped around double strands. So here, the topoisomerase comes in and gets rid of this extra, in this case, writhe. So let's just take another quick look at that. This is from our textbook, where we have, again, the helicase coming through, unwinding the DNA, untwisting the DNA. So the twist is what you have when the two strands are wrapped around each other. Writhe is when you have the double strands crossed across each other. You may see this in some of the exams. I'm not going to belabor this point. But pull those two apart, and you do literally get stalling that happens. This has been shown quite nicely experimentally. If you don't have a topoisomerase around, you can't unwrap this DNA any more than you already have. Yeah, sorry.
This might actually answer the next slide, but so does the topoisomerase attach further down from where the replication fork is to keep it from turning, or does it go directly behind or directly in front of where the helicase is?
OK, so the question here, just basically to paraphrase, is where does the topoisomerase sit relative to the replication fork? Because when you're pulling those strands apart, it's going to be ahead of the replication fork. And so as that polymerase is moving, it's going to be ahead of it. So it has to be ahead of that because that's where you're generating this extra, the writhe, which is happening with these things being wrapped around each other. So because you actually have two kinds of things wrapping around each other, you've got two strands that wrap around each other, and then double strands that are wrapping around each other. There are two kinds of topoisomerases that can take care of these problems. And it turns out they're completely interchangeable relative to each other. So two strands wrapping around each other are interchangeable with the double strands wrapping around each other. So these two kinds of topoisomerases, the really easy way to remember, topo 1 cuts one strand, topo 2 cuts two strands. So topo 1, oh, hang on, I've gone one too far here. Topoisomerase 1 will bind to a double-stranded DNA and make what's called a nick in one of these two strands. How does it do that? there's a tyrosine which is present in this topoisomerase type 1, and it basically pretends to be a 3' OH. So it binds to the phosphate, which is there in the backbone. So literally, it will cut, or also we say nick, because it's not gotten rid of any bases here, makes a nick, allows the strands to wrap back around each other, and then the reaction is reversed. And so the phosphate in the DNA will rebind to the OH, which is present at the 3' end. This is an isoenergetic reaction. So there's no ATP that's required to do this. And basically, the energy comes because your DNA is over-twisted or under-twisted. The other flavor of topoisomerase is topoisomerase 2, which takes those double strands, cuts them, and passes them through each other and religates them. Yeah, question?
So just going back to topoisomerase 1.
Yep.
So you're saying if it makes that nick, and because there's that stress of twisting on the DNA, it will just twist around that single point, come back around, and now the tension's gone.
Yeah, so that's exactly right. So since there's some extra tension which has happened because this has become either over-twisted or under-twisted, and we'll talk about that just in just a second here. But yes, that torsional stress, as soon as it's cut, then it'll go back to where it was, it'll be relaxed DNA, and then it will rebind to each other. So the energy is coming from that difference in twisting. And maybe it's a good time to move to that point is you've probably heard about negative supercoiling and positive supercoiling. So, and there was on that last slide as well. That's defined by positive supercoiling is twisting more in the direction that the DNA is twisted, and negative is going in the opposite direction. Each of those is going to generate torsional stress because you're not in the normal form. And we'll talk about the negative and positive supercoiling a little bit more as we continue through the rest of the class here. Okay, so the double strand, again, it's the same process. And this is, again, I left this up here to try and emphasize this. Is, if you think about me just twisting this, that's like changing one strand versus the other. It's just wrapping one strand more, and I'm doing a positive supercoil here because I'm going in the same direction as the helix. That then is going to generate these extra pieces which have flopped on top of each other. Your topo 2 would cut here and pass that through on the other side, which would be just the same as me untwisting over here. Now, that was positive supercoiling because I was going in the same direction as the helix of the DNA. On the other hand, negative supercoiling, what happens then? The same thing is going to happen. You're still going to get this wrapping around of the double strands around each other. Yeah. Pardon? This is writhe, where you've got these guys crossing across each other. And the twist is just those two strands wrapped around each other. Okay, that's all for for topology right now. We'll talk more about these different kinds of, excuse me, positive and negative supercoiling later on. We spoke briefly about this before. What happens to histones after replication? We talked about this when we're talking about heterochromatin being maintained after replication. This is another way of looking at that. You can ignore all of the names down here like CAF1 and NAP1. The basic message here is that as you undergo replication, those histones pop off because you can't replicate and you can't pull those two strands apart if they're bound to histones. But those histones will reassociate and reassociate quite rapidly after a replication fork comes through. You have new histones which will get associated with each of those strands, but you also have the old histones. And the key about those old histones, they still have that same modification. So if they were modified before, they stay modified. And that's how you can get your positive feedback loops taking place. Okay, so this is, again, just a diagram of looking at that. More questions? Time for me to ask a question. Oh, yes, before I ask my question.
So there's, after it's replicated with the two new strands, the old histones still have the modifications. So the new ones don't have the modification, or do they have to be modified
Right. So the question here is, do those new ones have modifications? Maybe I didn't explain that properly. The new ones do not have modifications. They've just been made. But they'll get modified in the same way as the old histones because of the read-writer complexes which are going to be there. OK, more questions. Yes? Delay the clicker as long as we can. Good. Yeah?
So I'm still just trying to figure out this. So I get that they don't have modifications, but then if they don't have modifications on a different strand, how is the reader, like, what is the reader complex basing? Because it needs to start at one that's modified, right?
Right. So, OK. Great question. So basically what we have here, the dark ones here, these parental nucleosomes, which, oh, sorry. The question was, where does the modification come from in the first place? Is that a good paraphrasing? Yeah. So any modifications, so pardon me, get this back, my pointer back here. Any modifications will be modified here in your parental nucleosome. And so those nucleosomes will come off, but they'll go back onto the DNA which is right next to it, because, of course, that's where your replication fork is. And so those will have those modifications. So whatever modification you have here already, some of those, for instance, here, there's a nice example, the dark ones. So maybe I should have mentioned, everything which is dark here represents the old nucleosomes. The light represents the new nucleosomes. So here, clearly, this is a completely dark nucleosome. It's going to have the same modifications as we had here. Yeah, so that's exactly right. So it's just like you see with DNA. If you're looking at how the nucleosomes are being maintained, it's semi-conservative. So each of those new strands is going to get half old, half new. It's not exactly like that, but that's basically the case. It's more stochastic, and so you don't always have exactly half and half. But it's going to be, on average, it will be half and half. Yes? So the question has to do, sorry, I'll paraphrase this again. What happens when you're getting closer to barrier proteins, particularly if we're talking about barrier proteins that are binding to nuclear matrix? That's an absolutely fabulous question and very astuteful. Is what seems to happen during replication is those are released from the nuclear matrix. Probably actually to some extent having to do with the topoisomerases, which are coming through and moving them off. I'm not sure that it's really well known how that happens. Okay, other questions about this? Okay, finally. Took us long enough, didn't it? Everyone's got your clickers out? Good. So which of the following has primer removing activity in Saccharomyces cerevisiae? DNA helicase, RFC, PCNA, FEN1, DNA polymerase 1. Ten, 5, yes, FEN1 it is. One thing I did want to sort of mention here, lots of people ask, what happens on the next exam versus the previous exam? Do I expect you to remember information from the first one on the second one? I would expect you to remember things like the fact that Saccharomyces cerevisiae is a eukaryote from one to the next, which is why DNA polymerase 1 and DNA polymerase 1 isn't present in eukaryotes. So, but everyone seems to be getting this. Good, nice. So I'll do really well in the next midterm. OK, so now I'd like to switch gears a little bit and talk about initiation. So yes, we've got our wonderful replication fork, but how do we get a replication fork in the first place? If you've got double-stranded DNA, how do you start replicating? So this is a very general case here of what happens in all DNA replication. Well, at least all cellular DNA replication. Viruses are different. Sign up for virology if you can on Friday. It's going to be an amazing course. There's also going to be a really fun lab course that's kind of associated with it. I'll tell you more about that next week. So what happens in terms of DNA initiation in cellular organisms is the first thing that happens, you have an initiator of some kind, and we'll talk about what it is. It's different in bacteria and eukarya, which binds to a specific place in your DNA, and it's very specific in terms of bacteria. A lot less so in eukaryotes, interestingly enough. And then with that binding, it is going to be next to usually a part of DNA which is easily melted. What do we mean by easily melted? It means that it's easier to pull those two strands apart. So what kind of bases do you expect to be in these areas? ACs or low GCs, either, both. So low GC, high AT. So these two strands come apart relatively easily. And then you have, what we have on here is protein recruitment. And the most important of the proteins to recruit here is this one that wraps around one of the strands. What's that going to be? It's wrapped around one strand. If it's wrapped on one strand, which one's that going to be? That's going to be the helicase, exactly. So the helicase, and particularly having the helicase pull those two strands apart, that is the critical part of initiation in any cellular organism. Getting those two strands apart. Once you have the two strands apart, then the primase can come in and do its thing, and you have extension of primers, etc. But getting those two strands apart, that's the really key aspect of this, and it's the helicase which is doing that. Yeah? Is there more than one helicase in general, in this image? So what happens here, and this is true in, again, all cellular DNA replication, cellular DNA replication, again, always exceptions here, is when you pull those two strands apart, you're going to have two replication forks. So you have a replication fork which is on this side, and your replication fork which is on that side. So you have a helicase for this replication fork that's going to go to the left, and this replication fork is going to go to the right. So you have the two helicases there because you're going to be what's called bidirectional replication. So it's bidirectional, semi-conservative, what was the other one? Semi-discontinuous replication. So bidirectional, semi-conservative, semi-discontinuous. Yeah? So the question is basically, if you don't have a helicase, how do you get a helicase on in the first place? Is that a paraphrase of your question?
Yeah, well, the strand is separated.
Right, so it's separated. How did they get separated in the first place? So one of those is the fact that it's low AT. The other thing is, guess what happens in most genomes? Probably heard about this. They're negatively supercoiled. Negatively supercoiled means they're unwrapped. So you're going to have, particularly in regions that are A's and T's, things that come apart with a certain frequency, a lot like nucleosome breathing. So there'll be enough of those, and then it's the binding the initiator protein, which brings then the helicase loader, which is really the main initiator interaction there. Because just like you have with a clamp loader to get a ring around your DNA, you've got to get a ring around that single strand. So you need helicase loaders as well. Yeah?
So that negative supercoiling, is it like it's putting a stress on that bond? Is that kind of what you're saying? So it's like easier to break them apart?
Yeah, once you've unwound the DNA, they're not going to be as stacked on top of each other, which is what's giving you your stability. So it's easier for them to come apart. Okay, so if we think about the E. coli genome, true for E. coli, not true for the elephant in this particular case, there's actually only a single origin of replication. So 5 million base pairs, as most of you got right on the exam, yay. Single origin of replication. So your replication initiator spine there, you have two replication forks that go all the way around the genome, and end up at the other end. People talk about this as the cellular origin of replication. We'll talk about plasmid origins later on. So ORI-C, it's the start place, places where things start on the chromosome, and it's where you have these initiation factors, which are going to bind, and then where the helicase gets loaded in the first place. And again, 2 replication forks. You can do the math here. It's actually more like, depends on your strain of E. coli. If you've got 4.5 million base pairs, 10 base pairs per turn, you've got to pull those apart. How many turns have you made? 450,000 that you need to get rid of with topoisomerases. So clearly, topoisomerases are very, very important. Turns out a bunch of antibacterial drugs, antibiotics, will target these topoisomerases. So they're really good drugs for treating, because it turns out those topoisomerases are different enough in bacteria from the host, i.e. us. So also really nice drug targets. This is just another way of looking at this. Here we have the replication origin. This is ORI-C in bacteria. The first thing that binds to it, again, creatively named DNA A. So it's the A piece. That binds to this initiator next to the AT rich. That then brings in DNA B, which is the DNA helicase. That helicase loading protein has to happen. And then we have DNA primase, DNA polymerase, etc. So again, it's the loading of that DNA helicase, which is the most important step in terms of getting our initiation. So this is what happens in bacteria. In eukaryotes, we have the ORC, and it has nothing to do with Lord of the Rings. It's the origin recognition complex. And whenever we talk about a complex, that means it's a multimer. It's got lots of polypeptides that are associated with it. Oh, hang on. There's one other quick thing about E. coli. So if you only have one origin of replication, if you are growing relatively rapidly, which is what E. coli does, you have to be able to recognize that origin of replication as something that needs to be replicated. And so this is true for ORCs too. We'll talk about this a little bit later on. But that DNA A complex needs to recognize DNA which hasn't been replicated yet. How does it do that? It turns out that DNA in E. coli is methylated. And that methylation happens on the DNA. It's actually in usually in the major groove of the DNA. That methylation happens well after you have replication that takes place. Since replication is semi-conservative, as soon as you start replication, you're going to have what's called a hemimethylated DNA. Now, hemimethylated, all that means is one strand is methylated, the other strand is not methylated. DNA A doesn't bind to hemimethylated DNA. So this is a mechanism for saying, hey, this has just been replicated. Don't bind here again. Don't start replication here again. Wait till it's all done, and then you can start replication again. This is also what's called licensing, and the licensing has to do with this is a license to initiate. So if it's methylated, it can initiate. If it's hemimethylated, it's not going to initiate. So what about eukaryotic origins of replication? This is a little different. One thing that hopefully all of you know is that eukaryotes use multiple origins of replication, not just a single origin of replication. And turns out that their DNA polymerases are way wimpier. They don't go as fast as the polymerases that you find in bacteria. So you basically have to have multiple origins of replication. And what's one of the other things that eukaryotes do that bacteria generally don't, or at least not in the ways we talk about it? Cell cycle. So S phase, M phase, etc. So there's only one part of the cell cycle where you undergo DNA replication. So eukaryotes have a big, lots of problems in terms of doing DNA replication. So it's considerably more complicated, but the same idea: initiator, recruitment of proteins, and helicases starting here. So you have your origin recognition complex, the ORC, which binds to origins. And so it's not like a single origin, as we have for bacteria, or at least E. coli. It's multiple different origins. Once you have binding of the origin recognition complex, then you have these two proteins, CDC6 and CDT1. CDC6 stands for what? What's CDC stand for? Cell division control, exactly. And so that has to do with replication. All of these things were originally found in yeast, most of them in Saccharomyces cerevisiae. And as the geneticists do, if you break something and try and figure out what you broke, you'll name that gene after what the phenotype is. So if you make a mutation in a CDC protein, you're going to have problems with the cell cycle. In this particular case, it's the sixth one of those genes, and it's important for initiation of replication. So you have CDC6, and CDT1 was one that was found a little bit later. All of these together will bind to the helicase. This is now called MCM. I didn't mention why it was called MCM earlier. MCM stands for mini chromosome maintenance. So maintenance of chromosomes. How do you maintain chromosomes? Because they're being replicated. So if you have a mutation in mini chromosome maintenance, you're not going to be replicating. So MCM, CDC, those then all to come together and sit on an origin and wait until S phase starts. So you've got your helicase there, it just hasn't been activated yet. How does that activation take place? You have the cell cycle. Cell cycle, lots of different things, but as far as we're concerned, it's all about the kinases. So you have a cyclin-dependent kinase, which will phosphorylate this CDC6. As soon as you have phosphorylation of CDC6, you also have phosphorylation of the ORC protein. Of course, all of these are kinases. They're protein kinases. They're putting phosphate groups onto things. Once you've phosphorylated the ORC, now you release the MCM. The helicase goes. Once you've gone helicase, you've got single-stranded DNA. Primase can come in, replicative polymerases, etc. Now, what you end up with is replicated DNA with a phosphorylated ORC complex bound to it. This phosphorylated ORC complex can't bind to CDC6 and CDT1 because it's got this phosphate on it. So this phosphate down here needs to be removed, going through to the gap1 phase of the cell cycle. Once you don't have this phosphate, then you have CDC6 and CDT1, which allow the helicase, etc. So this whole process here, how you can get replication, and replication to only be in the S phase of the cell cycle. Okay, origin, sorry, yeah.
Oh, sorry, what did you say CDK is?
Pardon? CDK is cyclin-dependent kinase. So in the cyclins, these are proteins that vary in terms of amount through the cell cycle. And we're not going to talk more about that, but in cell biology and hopefully in principles, people talked about them a lot. OK, so now we talked about the beginning. What about the ends? So I mentioned this a little bit already. If you have a linear chromosome, which we do, how many chromosomes do we have? Depends if you got them haploid or diploid. And it turns out that we have lots of different numbers. But we have all of these linear chromosomes. What's a problem with a linear chromosome? Problem with the linear chromosome, when you get to the ends, that since you only start with a RNA primer, that RNA primer is always getting degraded. Well, if you're starting with RNA primers, you get to the end of the genome, then this piece is going to get cut off. What cuts it off? The flap endonuclease, although that doesn't work terribly well at ends. What else? RNase H will be chopping off this particular piece. And so if you get to the very end of a linear chromosome, this lagging strand is going to be missing a piece. Well, now this piece is too short. If you replicate to here, this piece is shorter yet. You undergo another round of replication, this will get shorter and shorter and shorter and shorter. So what happens is the ends of your linear chromosomes are going to keep getting shorter. And it does appear that this is part of the problem that we have when we age, is that our chromosomes just get shorter and shorter and shorter, and then at some point gets into important genes. This is, in fact, maybe one of the reasons that we don't have very many protein coding sequences in our genomes, because at these ends of the genome, if you're chewing up a bunch of important genes, then you clearly have some problems. There's one really easy way to deal with this, and that's what most bacteria figured out. Don't bother with ends at all. Have a circle. Way easier. Now, you have issues with that as well, and probably you can only get to a certain size of just having a circular genome. And having these linear ones probably allows them to get considerably larger. So what do? You have telomeres. Telomeres are made by a really fascinating enzyme. It's called the telomerase enzyme. The telomerase enzyme includes an RNA and a RNA-dependent DNA polymerase. So a reverse transcriptase, potentially derived from some of the viral reverse transcriptases as well. So what does a telomerase do? All that a telomerase does is makes repeated sequences at the ends of linear chromosomes. And how does it make those repeated sequences? It makes those repeated sequences from an RNA template. So here's this RNA template, which is always part of the polymerase, both the RNA and this enzymatic activity here. It actually, obviously, you can tell here, it looks a lot like a DNA polymerase, right? It's got fingers, it's got a thumb, it's got a palm. Believe me, it actually does in three-dimensional structure. But if you just look at the structure of one of these things, you can say this is a DNA polymerase, because it's got that right-hand structure to it. So it's a DNA polymerase, but it uses an RNA template. Again, it's reverse transcription. So what does it do? It just literally adds repeats. Well, it's only got a relatively short piece of RNA that it copies. So what you end up having lots and lots of repeats of the complement of this sequence, which is right here. So multiple copies, what does that do? It protects the ends because you've got now a much longer end as opposed to losing one each time. Then you can keep adding on more and more. That will be replicated. Also because you now have this repeated sequence. This repeated sequence on both strands can bind to itself. So not only do you have an extra length that through these lagging strand polymerases and the primers which you're getting rid of, those are still okay. But you can also form this base pairing interaction. Turns out also lots of proteins that are involved there. Now, why is that? I didn't mention the other problem that you have with ends. Ends are really yummy. What I mean by yummy? Exonucleases. There are lots of exonucleases present in cells. And so if you have free ends of DNA or RNA, exonuclease is going to chew on those like mad. So it's not just missing primers, it's also the ends of your DNA. And we'll see a little bit later that this also is involved in DNA repair processes. So this is an overview of that. I mentioned these loops. These loops are happening because this sequence here is going to be complementary to the other sequence here, and that's because your telomerase will have added onto the end of your DNA. How does it add on to DNA? As I mentioned, this is a DNA polymerase. What is the activity of a DNA polymerase? It extends what? 3' OHs with a template. The template here is RNA, but it's still extending 3' ends. And what that means is it's actually just extending your template strand. You're always going to have a little piece here. So here's your RNA primer in green, which will be extended with DNA polymerase. The telomerase will make an extra copy. You'll have another primer, which will extend here. But you're always going to have this little piece at the end here, which now, 5' to 3', GTTG is now complementary to this CAAC here. So it can wrap around. This piece can base pair with this piece of DNA, and that's exactly what happens here with these so-called T loops or telomerase loops that you'll see at the end of DNAs. There are many other proteins that are associated with this. You do not need to remember these. I can't remember them. It becomes ridiculously complicated. We actually tried to hire a faculty member here in the biology department who works on some of these things. Really fascinating process. All of this extra stuff. If you're interested in telomeres, probably also really important for aging, check out some of these links here. Another way to look more at telomerases, sorry, there's one more thing here, is a really nice lecture by Liz Blackburn, who got a Nobel Prize because she was the first one to characterize telomerase and the reverse transcriptase. Excellent talk, and I'll have a link after the clicker question. So if a telomeric sequence is 5' CAGGC, the sequence of the telomerase RNA must contain from 5' to 3' CAGGC, GCCTG, GCCUG, GUCCG, ABCDDFG. Stedman's got to come up. Oh, hang on. We haven't started yet. Come on. Start. Yes, OK. Now all of you know exactly what the answer is, right? E. Ten. OK, so CAGGC, right? So always anti-parallel to each other. So here's our 5' here. What do we have? G, C, C, U, G. And it took me years to figure out how to draw these things upside down. So, but you can also just flip it over. That works as well. So what's our answer? D. C. Sorry, C, not D. So that's why I have to write these things down. Otherwise, I get them incorrect. Yeah? No. Yeah, your question? So the question is here, if the telomerase is extending the template strand, that's the template strand that was made by the DNA polymerases to start with. The template that the telomerase uses is the RNA, which is part of the telomerase enzyme. So it's copying that RNA. And that RNA then will extend at the 3' end of the... Yes, exactly. So what's happening there is it's copying its own, because there's no template for it to copy from at the end of the genome, because that's the problem that you're falling off at the end with. And so it's got to bring its own template along with it. Sorry if I didn't make that clear earlier. Yeah?
I'm kind of curious how the T-loop forms. You might just be able to go back to the slides and explain this to me, but the sequence isn't like a complementary construct to itself. It's the T-loop forms from, I guess, what?
So the way a T-loop forms is this sequence at the end here that the telomerase keeps adding. This now will get copied. Let me hide this now. Excuse me. What do we do with our... Now you'll have the cellular primase and polymerase, which will make copies of this. And so it's now going to make a complement of this strand. If you think you've got hundreds of copies of this over here. And so you've made complements of this sequence. So you have a complement of the sequence. You're still going to have a little piece at the end here. For instance, this GTTG, which is at the 3' end. But the telomerase has also made all of these GTTG copies right here. The opposite strand is going to be C. It's actually a little bit offset here. CAAC. CAAC is complementary to GTTG. So it displaces actually the opposite strand. So you end up with a loop with one strand and then that single-stranded piece that comes into it. We'll talk more about single-stranded pieces when we talk about DNA repair at this point on Monday. So, yeah.
Just for clarification, the RNA sequence of the telomeres, those don't change. They can't be modified.
Yeah, so the question is here, what about those sequences of the telomerases? Do those sequences change? No, that's a sequence which is present with the telomerase enzyme. And so that's always going to be it. So if you look at the telomeric sequences of every single one of your chromosomes, they're going to be the same because they're going to be the complement of that telomeric RNA. It does change a little bit from organism to organism, but in one organism, you're always going to have that same telomeric repeat.
So does that mean there's multiple different types of telomeres just shuffling around, looking for a specific sequence for the template?
No, it's one telomerase, but many, many copies of that telomerase enzyme. So every one of the ends of your linear chromosomes will have a telomerase that associates with it, but you have that one telomerase gene and one telomerase RNA gene, which is always going to be the same. So it's always going to be making those copies. Does that sort of make sense? In the back, yes. The question is what happens when you age? Sorry, I'm just paraphrasing here. What happens when you age? So what happens when you age is that telomerase activity, in fact, slows down as you age. We don't quite understand why that is. And a lot of people have talked about, how can we deal with this? One other thing, and one of the reasons that people are really interested in this, is it turns out that in a lot of cancer cells, telomerase gets upregulated. And so, because if you're replicating like mad, which is what happens in cancer cells, you're going to have exactly the same problem. And you keep losing ends, then you could potentially have a problem. So this is also a target for doing anti-cancer work. Yeah?
What if you have a chromosome that has mostly heterochromatin on the ends versus euchromatin?
So the question has to do, what about euchromatin versus heterochromatin? Sorry to paraphrase again your question, but when we're undergoing replication, the whole chromosome gets replicated. So heterochromatin actually does form at telomeres, which does shut those down, but that's after replication and after the telomerase has had its activity. That happens later. Okay, so I mentioned there's a really nice telomerase review here. This is this iBiology seminars. A number of people have asked me about what are good sources for looking up various different aspects of different parts of molecular biology. iBiology.org, completely freely available, really great seminars. And this particular one, Liz Blackburn, she talks about telomeres and telomerases and the whole discussion of this. So finishing up with DNA replications, this is a real quick review. We talked about all this stuff last time. That was the midterm, so we've forgotten it already. And today, we talked a little bit about origins of replication. That's part of what we call a replicon. Replicon has an origin of replication, also a termination of replication. How replication terminates is actually a really interesting question that we don't completely understand. So the polymerases run into each other. What happens then? I'm going to do a quick lead-in to DNA repair right now. Got a few more minutes before everyone's going to run away for the weekend. And just wanted to go over this very briefly. DNA is a horrible molecule in terms of genetic information. And that's partly why people thought that proteins were a much better way to carry genetic information. And that's because every single part of this molecule can be modified chemically. And it's that chemical information in terms of the base pairs and how your double strand is associated that's absolutely critical for passing things along to the next generation. So you have all of these changes that can happen. Once you have a change, particularly if that change turns out to change what gets added to it when you replicate, that can get passed along from from generation to generation. So mutations can be really bad. They can also disrupt replication. So you get to a certain point. The DNA polymerase, as we talked about before, it's all about structure. The DNA polymerase is getting its fidelity by showing that you've got the right double-stranded piece. It's the right distance between those base pairs, which, by the way, was the answer to the wrong base pair that I had up on the slides last time. I'll bring some extra copies of that later if people want to chat about it. Also can disrupt transcription. Transcription is also very dependent on the structure of your double-stranded DNA. Probably lots of these mutations are important for cancer. Talk more about that later. Maybe the accumulation of mutations above and beyond what happens with telomeres is important for aging. And as we'll see, we're having mutations happen all the time in our genomes, literally while we're here. So how do we deal with those? There are a couple of ways that that gets dealt with. Probably the best way to deal with mutations and changes that happen in DNA is we have a backup copy. We've always got a backup copy around if you're a double-stranded piece of DNA. So if there's a mutation that happens over here, your information over here is a way you can correct it. That saves us in many, many different ways. Now, of course, if you didn't have mutations, we wouldn't have evolution, and we would probably all be little puddles of slime on some planet or other at some point. So wanted to mention some of the reasons that people are interested in DNA repair. These are just a few of the genes involved in DNA repair in humans, and they lead to all kinds of things. Important here, you'll see cancer, 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 leukemia, etc. So many of these proteins are involved in DNA repair, and when they're mutated, are giving people, and in this particular case, lots of these are inherited diseases. So if you've inherited, unfortunately, one of these particular genes, their allele of that particular gene, you're going to have very high rates of cancer. And these turn out to be important in all the different kinds of repair, which we'll be talking about here in just a second. And I'll just talk about how we get mutations, and we'll finish up here. We'll talk about how to repair them on Monday. How do you end up with mutations? One is why you don't want to be too close to places like Fukushima. Ionizing radiation. Ionizing radiation is actually a really huge problem because what it does is it actually ends up breaking both of the strands of your DNA, so you don't end up with that extra backup copy. So if you're normal and in a good position, that's great that you would have that extra strand, but this double strand would be really bad. On the other hand, if you're a radiation oncologist trying to treat a cancer, using radiation to break double strands is actually really good, and in fact works quite well in a number of cancers. But fortunately, most of us are not exposed to ionizing radiation most of the time. On the other hand, we are exposed to heat, and when I say heat, 37 degrees C, which is about what I have now. We like oxygen, and we like light. Unfortunately, all these things are really bad for DNA. Deamination is one of the most common. This is a base modification that happens mostly, but not exclusively, on pyrimidines. Deamination of cytosine is one of the most common. The most common is depurination. So at 37 degrees C, between 5 and 10,000 of your purines fall off the DNA. Uh-oh, that's like way higher than our, what do we have, 1 in a billion, 1 in 10 billion in terms of a replication process. And then finally, don't go out without sunscreen because what happens as soon as you have two pyrimidines that are next to each other and they're exposed to UV, they will cross-link to each other. And again, just a chemical reaction causes all kinds of problems with your DNA. So fortunately, in Portland, we don't have to worry too much about UV radiation. Certainly this weekend. I, again, highly recommend the Festival of Lights, and we'll talk about some of these changes on Monday. Have a good weekend.